Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is Thursday, October 13th, and we're talking to Mel B. Feany, professor in the Department of Pathology, the Division of Neuropathology at Brigham and Women's Hospital in Boston and Harvard Medical School. Mel studies the genetics of neurodegeneration and uses the wildly successful fruit fly preparation to analyze the connections between genetics, cell biology, and disease phenotype. Actually, she does a wide variety of other interesting things as well, but I think that's what we're going to be talking about today. So welcome, Mel.
Thank you. Happy to be here.
With us, we have two UTSA experts on neurodegeneration, our own Hyoung-gon Lee.
Hi.
And Chris Gamblin.
Hi there.
Mel, it seems to me that explanations of alpha-synuclein pathology in Parkinson's disease, the mechanism always starts with some overexpression or maybe misfolding of synuclein, and then some unknown number of steps in between, some gap in what we know, and then some series of things that go wrong, like mitochondrial failure or autophagy or cytotoxicity or something like that. And then, or all of those things. So maybe all those things are different pathways, and each one of them is a different effect of alpha-synuclein, or maybe all of those things have some common denominator. And in your work, it is implicated that actin cytoskeleton as possibly a common denominator in the mechanism of cell dysfunction in Parkinson's disease and maybe some other neurodegenerative diseases. So could you start just giving us an overview of what this grand unified view of the causal link between synuclein and cell pathology?
Absolutely. I'd be delighted to. So as you mentioned, we do use the genetic model organism Drosophila, and one of the things that Drosophila has been really terrific at is identifying a cascade of events where one thing comes first, the next thing comes, and then another thing comes in a more or less linear pathway. Of course, biology isn't always going to be perfectly linear, but what genetics does allow us to do is to try to order events. And so we have been working to order events and, more importantly, probably understand the players that are involved in each of these events. And our idea, based again on genetic gene finding experiments in Drosophila, is that the protein alpha-synuclein, which is a key player in Parkinson's disease, binds directly to the protein beta-spectrin, which is a very important organizer of the plasma membrane of the cell. We think then that through beta-spectrin, alpha-synuclein indirectly influences the actin cytoskeleton, and the actin cytoskeleton has a number of important roles to play, but we would say one of the more important roles is to control the division and ultimate health of mitochondria. And when mitochondrial health declines, that then we think leads fairly quickly to neuronal cell death. So that's sort of a simple way we would order events based on our experiments using Drosophila.
So what's going wrong with actin? I mean, so maybe you say something about dynamic and stabilized.
Right. So what we know in general about actin is actin-dependent processes require dynamic actin. So that requires, actin, you can visualize actin as a filament, if you will, and these actin filaments are used in the cell to move organelles for structural stability, but it does appear that all of the functions of actin that we know about require that actin have some dynamics, that it becomes shorter, that it becomes longer, and that helps with, active maintenance of cell shape, and it helps move organelles around. Or in the specific case of mitochondria, we think it helps recruit specialized proteins, particularly DIRP1, that help mitochondria to divide, which is a critical aspect of mitochondrial biology. So we believe it's really the dynamic nature of actin that's required. Now, A lot of the work we do, however, is in vivo preparations in the fly brain or in the mouse or rat brain or even the human brain, where it's harder for us to look at the actual dynamics of actin. So what we often do is look at a sort of a single time point, how stable actin is, how much is in filaments, how much is in monomers. And so we think, and so then so what we observe with alpha-synuclein in alpha-synuclein models of toxicity, that there's more stabilized actin. But we really think that the fundamental problem is likely to be that stabilized actin is insufficiently dynamic. It can't respond to challenges and changes in the cell.
So you see it redistributed in the cell. The actin would have been here and now it's there. Is that?
Well, maybe. I think we would view it in a little bit of a different fashion. I think we would view that we should have, let's say on the mitochondrial surface, that we should have an actively turning over pool of actin, but rather what we see is an excessively stabilized pool of actin.
So you can see that just by measuring how long it takes for actin to rebuild itself? Or
that would be ideal. We would love to do that. Those are experiments we really like to do in cell culture, but we haven't done those yet. But that would be the kind of experiment that one should do to really prove that point.
So how else could we measure the dynamic versus stabilized actin?
You need some kind of more live imaging. So the indirect measures, what we have done in a lot of the data that I showed to the group earlier today, which is to simply measure the amount of F-actin or G-actin.
Mm-hmm. I see. So F-actin is formed into filaments and G-actin is monomeric.
Yeah.
So if there's a lot of dynamic actin, then there should be a lot of G-actin available. I'm just trying to get in my simple mind.
I'm not sure that necessarily numbers, more of this or more of that. I would say it's more that the balance has changed in an appropriate way.
So how much does it change from one neuron type to another, or even if you were to go into a non-neuronal cell type? I mean, is the distribution of actin about the same? Is it different?
That's actually a fascinating question. I do not know that we know the answer to that. That would require either isolating those cells or performing some kind of live imaging to look at dynamics directly. And I don't know how much of that's been done. We have not done that. That's a really interesting question.
Okay.
Yeah.
So I understand how actin could be important for mitochondrial division. Why is mitochondrial division important for the cell?
Well, that's a great question. And I think that's not a question that's fully answered, but I think probably the leading hypothesis for that is that the way that you maintain a healthy balance of mitochondria is mitochondria divide, and the mitochondria that that are less functional are selectively cleared. I think that's everyone's favorite model about why mitochondrial dynamics is so important. And it is very important. So others, in fact, people at this institution have shown that if you prevent normal mitochondrial dynamics, normal mitochondrial division, that's very toxic to the cell. So that's really clear, I think. Why exactly, I think is a little bit more of a hypothesis that's needed to clear dysfunctional mitochondria to maintain a healthy mitochondrial population.
I see. So you'd have a bunch of broken mitochondria hanging around. Can you see that?
Yeah.
I mean, can you see that the mitochondria are broken?
So the data that supports this idea that the reason that mitochondrial fission is important is to maintain a healthy balance of mitochondria is data that shows if you either interfere with mitochondrial fission or you interfere with mitochondrial clearance, then you have a population of dysfunctional mitochondria that persists and sort of takes over the cell, if you will.
And the story that mitochondrial dysfunction is really important in Parkinson's disease goes back a long way.
A long time, yes.
Has a big
absolutely
history.
Long history.
But but I've never heard really a clear statement about what it means for the brain cell. I mean, we see neurons die in substantia nigra. Do they die because their mitochondria can't produce enough ATP, or?
Yeah, so that you're asking a lot of really important questions there. So I'll start by answering the ones that I know the answer to.
Good.
And then I'll confabulate on the later ones. The mitochondrial theory of Parkinson's disease and of Alzheimer's disease and of every other disease, it has a checkered history. So if you look in any of these disorders, including more that we haven't explicitly spoken of, you'll typically find abnormal mitochondria. But I think, and you guys can, these stories very well too, you can certainly chime in, but my feeling is that until there were genetic mutations that specifically implicated mitochondria, in the case of Parkinson's disease, it's mitochondrial dynamics, and in the disease process, I think that there were really schools of thought that said, you're seeing these mitochondria, they're definitely abnormal. Yes, we believe that, but that's because the cell's sick, right? Uh-huh. As opposed to the cell dying because the mitochondria are abnormal. To me, and you guys have, I'm sure, your own feeling about this, to me, the turning point was finding genetic mutations, and particularly in common diseases like Parkinson's disease, where those genetic mutations clearly implicated mitochondria because they were mitochondrial proteins and they were important for mitochondrial function. Before then, there were diseases like primary mitochondrial diseases where the genetic mutations were in genes that encode mitochondrial respiratory chain components, for instance. So, of course, you knew in those rare diseases that mitochondrial dysfunction was the problem. But I don't think we really knew in common diseases that it was a cause rather than a side effect. But you may have your own take on it.
So the thing that always concerns me is when you're looking at mitochondrial dysfunction, I feel like it can cause an increase in oxidative stress, but it can go the other way as well. The oxidative stress can affect the mitochondria.
Absolutely, yeah.
And so the thing that scares me in trying to understand diseases is how possible is it that it's going in one direction in one individual and going the other direction in another individual, but they both have increased oxidative stress and increased mitochondrial dysfunction, but which one caused which, and can it be different?
Well, that's the problem with common diseases like Parkinson's and Alzheimer's. You emphasized and I emphasized earlier genetic causes, but what neither of us said is that most people with Parkinson's and Alzheimer's don't have single gene penetrant mutations that are causing the disease. They have important genetic influences, and we understand a little better through GWAS those now, but still they're at some level sporadic. And so what you say could exactly be true. Things could be different one patient to the next. But that's going to make it really hard to do science on them. But maybe you have to take that challenge on.
So mitochondria do a lot of different things. So you were just mentioning oxidative stress. Maybe you were thinking about reactive oxygen species, and which can be directly damaging to the cell. That's definitely one way mitochondria could harm the host cell. There are others, though, like calcium disposition is very important.
And even, with oxidative stress, we tend to talk about that as being something negative. But of course, there's important signaling that goes on with reactive free oxygen species, that's very important in normal biology.
In addition to that, I think another prevalent view is that it's not a gain of toxicity. It's basically about the fitness of the mitochondrial activity. Eventually, it's going to be cell, neuron fitness, like mitochondria, even though it doesn't increase the ROS or other toxic molecule, like it lost normal function because of that the dynamics is broken or some structure is broken, pathway is broken. It cannot function well supporting the neural function, for example. So then the consequence is that it's going to be more vulnerable to the additional stress. That's what we my group is actually previously to talk about, like what we call the 2-hit hypothesis. So it's going to be a more vulnerable. Some stress is usually okay, so it can endure, but eventually it's going to take the hit and then go to the degeneration. because of, cells are more vulnerable because of their mitochondrial activity, doesn't maintain their cell neuronal fitness well.
So it just weakens
right,
the
yes,
cell.
weaken the cell, right.
And then the thing that actually kills it could be anything.
Right.
Wow. That's kind of distressing from a scientific point of view. Because
it's the multifactorial hypothesis of these disorders, which has almost got to be true at some level.
I think that's what, we can see a lot of heterogeneous, multiple event or different pathway related to mitochondria. We can see in that model study because of that kind of thing is more complicated as we are kind of talking about it, because it's so multiple event, happen in different cells, even the same animal probably, or same patient. So I think it's not just a one-way explanation or one single mechanism can explain that how the mitochondria contribute to the pathogenesis, for example, like Parkinson or the Alzheimer's disease. So that's what I'm thinking.
Well, you mentioned some other things that are affected by actin that also sounded like they could be deadly for the cell, like control of the sodium potassium ATPase.
Mm-hmm. Right.
Which seems independent of the mitochondria, at least by mechanism, right? So actin is somehow...
Yeah, so, it's interesting you bring this up, and maybe this isn't exactly what you want to talk about. You can stop me if that's true, but, I think we, originally got on to actin through the genetics from the fruit flies, as we mentioned, and, we thought that would be an important contribution to just characterize what was happening with actin. If there were changes, that would be good. If we could show those changes were meaningful for the toxicity, we thought that would be a contribution. Where we expected we'd really run into trouble, and I sort of thought we might never really get anywhere, honestly, is because actin does so many things in the cell. How are we ever going to show that any particular downstream target was important? Mm-hmm. But fortunately, we didn't let that stop us. You might think that, and you're just like, okay, I'm not going to tackle that. But I'm really glad we didn't stop there, because in fact, it was possible to show that particular targets are important. Are they the only targets? Probably not. And you saw from the talk today that, we're thinking beyond a particular target, but the first target we identified was mitochondria. And again, with the power of genetics, we can say, it is a target, and it's a meaningful target, because when we correct that particular target, the animal gets better. So it's not the only target, but it's an important target. And I think that's one of the things genetics really allows you to do. So probably there will be other important targets. I didn't show you too much of this. In fact, maybe not even enough for you to really get the story. But a second target of actin that we've shown is autophagy. So we've shown that actin directly controls autophagy, and that the deficits in autophagy that occur in at least the Parkinson's model, perhaps also an Alzheimer's model, but at least in the Parkinson's model, are through actin. So if we correct the actin defect, the autophagy defect is also corrected. So again, a second target. And we showed that was directly through autophagosome maturation, and actin's known to control that process. So there's two targets that we can see. So we're never probably going to get to all the heterogeneity and all the actin targets, but I think we can make some meaningful progress on the major ones.
Mm-hmm. To me, that is something very satisfying about the ubiquity of actin, and because as an outsider, I listened to that cell biologist talking about diseases. And you talk to a cell biologist who's interested in autophagy, and all the diseases are autophagy. Talk to somebody who's interested in mitochondria, all the diseases are mitochondria. And it seems like every target that anybody studies is a target that seems to do something. And you go, wow, it's just like whatever you study, whatever you look for, that you find it there. And so it makes me a little cynical. But in this case, if it turns out that it's the cytoskeleton, then it's not surprising.
So that is exactly what we think. We think that you're absolutely right. You look at the patient tissue, and we talked to how so important it is to go back to the patient tissue, as limited as it is for causality. But if you look at the patient tissue, mitochondrial abnormal, autophagy is abnormal. And so it is really very satisfying to identify a pathway that's controlling both of those processes, because it gives you some sense that you're starting to understand why things are happening. And then, of course, with the experimental model organism, you can say it's causal, which I think is critical.
So maybe it'd be good to dig in a little bit farther into the mechanics of these molecules. So it's not just actin, it's also a couple of other cytoskeletal molecules. And in fact, alpha-synuclein doesn't immediately bind to actin.
Well, we thought it might. People had suggested that, but we didn't see that. So our hypothesis is really that the actin effect of synuclein on actin is somewhat indirect, although only through one additional binding partner, and that's beta-spectrin.
Yeah. So what is beta-spectrin?
So spectrins are large molecules that one thinks most about these large networks under the surface of the cell, the subplasmalemmal network, and we think about them mostly traditionally in terms of maintaining the integrity of the cell, the shape of the cell. And then, but they do other things. And so one of the important things that they do is bind proteins like ankyrin, and through ankyrin, also some of the details of this are a little bit unclear exactly who's binding to who. But in general, what we know is through the sort of the scaffolding that spectrin makes, the binding of ankyrin, and then the subsequent binding of that complex to plasma membrane proteins, that two things are important there. One is the compartmentalization of the plasma membrane proteins, and the other thing is that the recycling of those plasma membrane proteins is controlled. So those are some of the major things we know that spectrin does.
So compartmentalization means that they're in the right place.
In the right place.
They're next to
They're where they're supposed to be in the plasma membrane, and the right amount of them is on the plasma membrane as opposed to getting taken up into the cell.
So is there any indication that in synuclein might mess that up, and the molecules on
that's why that initial observation that alpha-synuclein is binding to spectrin made us ask the question, okay, so we know some of the things spectrin does, let's go look at those things, let's go look at the proteins that ankyrin anchors in the plasma membrane and look at the distribution. So we looked at the distribution of one of the major a major plasma membrane protein of known function, sodium potassium ATPase, and we saw that protein did not maintain its normal localization when we expressed alpha-synuclein. And also the function of that protein was abnormal in that the membrane potential of the cell was altered.
That seems catastrophic for neurons to have that.
Well, the neurons are in bad shape in people and flies, and to some extent in cells from patients that we grow and culture to better study the process. Yes, it's catastrophic. The people with Parkinson's disease have catastrophic loss. The neurons die. You can't get any more catastrophic than that.
So how much are these processes involved in just the actual neurotransmission and release of neurotransmitters?
Yeah, so I think that's really interesting. Of course, with alpha-synuclein, the alpha-synuclein is a synaptic associated protein. It's closely in the same place as synaptic vesicles, although it probably doesn't necessarily bind directly. And people have different ideas about that. I mean, some people think, as you guys know, that Parkinson's disease is a synaptopathy, and that the fundamental problem in Parkinson's disease is alterations in transmitter release. Mm-hmm. Some people feel strongly about that. There's a certain appeal to that idea because that's what synuclein normally does. I think our data has tended to suggest a little bit more of a gain-of-function mechanism in the sense that alpha-synuclein gets mislocalized from the synapse where it normally is to the cell body where it starts to impinge on localization of the sodium potassium ATPase or, as we discussed earlier, the various submembrane regions that are defined by different spectrin isoforms in human neurons and mitochondria. But I would say one thing that really does intrigue me about this new interaction that we've discovered about alpha-synuclein and spectrin, there is actually a pool of spectrin at the presynapse. That's not where people normally study spectrin. People normally study spectrin postsynaptically. But there are a couple of reports that have shown pretty convincingly, I think, that spectrin's at the presynapse. So I think it's a very intriguing question as to whether that interaction of alpha-synuclein and spectrin contributes to the normal function of alpha-synuclein in the presynapse. The total speculation. And that brings me to a larger hypothesis that I've had for quite some time, which is that it is abnormal interactions with normal interacting partners that underlies disease pathogenesis. So I would speculate in the case of tau, one of the things tau, you can get into this with me because you know tau, but I'm going to speculate that one of the things tau normally does is actually act in vivo as an actin cross-linking agent. It has some physiologic function, although I don't know what that is. Of course, it's hard enough to know what the microtubule physiologic function is of tau. But that normal function of tau is being co-opted in disease states into abnormal actin stabilization. And similarly, I think it's a really interesting idea that alpha-synuclein normally does interact with spectrin. So although we know where alpha-synuclein is, it's been really hard to understand exactly what it does in normal physiology. Maybe that's because it's interacting with spectrin, and maybe that's the key to understanding the normal physiology. But that process goes awry when, in disease states, alpha-synuclein is relocated into the cell body, potentially into the axon, and starts to interfere with spectrin interactions there, which it's normally not a dominant partner in those different localizations in the cell.
So in this kind of way of thinking, the Lewy body is not really part of the pathogenetic mechanism.
Again, that's highly debated. Our data has always tended to suggest that Lewy bodies are large sinks that are protective, that may be soaking up abnormal forms of aggregated alpha-synuclein, but that's a topic that's highly debated in the field. Many people would take issue with that and have other opinions. But I think, to me, it's always been hard to understand, you guys can get into this with me if you want, how that huge structure is really in a meaningful way interfering with the normal biological functions of these proteins. But I know not everyone feels that way.
So I've always looked at it as both protective and eventually toxic. So, with tau and neurofibrillary tangles, you can imagine that taking the bad tau, polymerizing it, and sequestering it somewhere in the neuron keeps it from doing bad things. But the neuron can only take so much of that. At some point, it just starts to occupy all of the space and the other things there.
And potentially soak up things like proteasomes that the cell needs. Yeah. So there's many potential mechanisms.
So I think that there's every possibility that early on in the process, aggregation is completely protective. But if it's allowed to go on for too long, it can have detrimental
doesn't need to be
effects.
One way or another, certainly.
Right. And part of the problem, though, is when we look at the brain, oftentimes we're looking at the survivors. And so the ones where tau killed the neurons, they're not there anymore. Yeah. So there's nothing to look at. Unless you're lucky enough to find a ghost tangle or something like that.
Right. And there's been
a ghost tangle.
What's a ghost tangle?
A tangle that remains after the neuron dies, so there's no neuron
left.
Yeah, and there's been some data from people like Brad Hyman, the numbers aren't large on this, but to suggest that when you can look dynamically in animal models, there is some evidence that those neurons that have tangles are actually doing better than the ones that don't. Yeah.
So in your fly model, can you see the Lewy body or?
Yeah, so one of the things that's been great about the fly model is I don't want to call them Lewy bodies, because I don't want to offend anyone, but they really do have inclusions that have all the things you'd want to see in a fly for a Lewy body. They're really fibrillar. The fibrils are the right size, and they have other components that people have known for a long time that Lewy bodies have. So there's been a recent flurry of interest in the idea that Lewy bodies not only have the alpha-synuclein fibrils, but they have other membranes, components of mitochondria, which of course the EM people knew for years and years and years. Mm-hmm. And so the flies recapitulate that. They seem to, have some peripheral membranes attached to them. So they do have very nice inclusion bodies.
So can you like track or make them some clear view that their chronological sequence that
how... So flies, of course, I love them and I like to think they're great for everything. The brains are pretty small, so to do dynamic imaging, that's kind of hard in the fly brain. Yeah, especially for those small inclusions. Okay. I think we maybe better leave that to the mouse people.
What's your idea? It seems to me that like Lewy body is more like consequence, end stage of the whole process.
A marker, yeah.
So process you just mentioned, interaction with the actin spectrin, and then eventually the actin processing problem. That's kind of likely early event, actual causal mechanism, and then as a consequence, that Lewy body can be formed, or it's going to be, doesn't matter. Actually, it's separate event.
Well, we don't think it's separate, and at least in experiments, when we rescue toxicity, there's more Lewy bodies, more inclusions, and they get larger. Oh. So that's part of the reason that we've always viewed them as potentially a protective sink. Although, as Chris says, there's no need to see it one way or another. Both things can happen at different times.
There's also a notion that the sort of fibrillary form of synuclein can propagate from one cell to the other.
One of the more.
Is this something that you have investigated, or you have?
So that's a hugely important topic for many, many investigators in Parkinson's disease, but not something we work on. We were interested to see if we would see that in our flies. We did some experiments and wasn't obvious that was occurring. The propagation experiments can be challenging to interpret because you have to make sure that what you're seeing is actual propagation as opposed to simply a little bit of expression in the cell that you're arguing is propagating. And we could never really convince ourselves of that. So we don't have good evidence for that in the flies, but I'm certainly it could occur.
Anyway, you're overexpressing kind of sort of generally and universally, right? So
what we did, and of course for those experiments, is we would, where we tried to see if we might see it, is express only in one cell population and then look at the synaptically connected cells or other surrounding cells and see if we saw propagation.
Well, that seems like a really good experiment, a really good test. You're not confident?
We didn't see it, but but it also isn't that easy to see necessarily. So people in mammalian systems had to work hard to see it. We may not have worked hard enough.
Okay, well, thank you very much.
My pleasure. Thank you for having me.
And Chris, and Hyoung-gon.
Thanks.
And this has been Neuroscientists Talk Shop.